My name is Neil, and I'm going to tell you about the Pseudomonas project that's available in Biology 2002. One way to start thinking about this project is to ask why there are so many different kinds of organisms in the world. Now that may seem like a strange question to ask at first, but people think about evolution as a competition that results in survival of the fittest. But when you look around at the world, you see all different kinds of organisms, like trees and fish and birds and insects, and they can't all be the fittest. And it's more complicated than that, because even within the category of trees, you have all these different kinds of species. Within the category of fish, you have all of these different kinds of species. Now, you'd think that if it's survival of the fittest, there would be just one species of fish, and the whole ocean should be taken over by that one species. Now, the Pseudomonas project is going to start with an experiment that addresses exactly that question by setting up a whole ecosystem in a little tube like this with a few milliliters of media, and then watching the population evolve. Now, a tube like this can easily contain billions of bacteria, which is more than all of the people on the planet. And so you can ask ecosystem scale questions and address them with laboratory scale resources. So working with this project can allow you to ask questions like things about invasive species or things about disturbed ecosystems. But it also turns out that one of the ways that Pseudomonas behaves in a system like this is to create a system called a biofilm. Now, biofilms are a growing topic in microbiology, and they have medical applications and biotechnology applications, and you'll get a chance to explore how these systems work. Now, there are advantages to using a low-maintenance system like bacteria. So, for instance, if we took dogs and put them in an enclosed space and forced them to fight for survival, that would be cruel and we would go to jail. But we can do experiments like that with bacteria and it's no problem. So in general, if you plan ahead and communicate, it's very easy to break these experiments up among the different members of your lab group. And so you get a chance to develop and practice some of the soft kind of teamwork skills that are valuable in any lab or workplace. So in general, the Pseudomonas experiments are going to emphasize conceptual thinking over a lot of complicated hands-on skills. Once you've learned how to start cultures, how to keep them from getting contaminated, and how to plate them out in petri dishes to examine the results, there aren't a lot of other specialized skills to learn. So what you'll need to do is to think very clearly about the questions you want to ask and about how the data is going to address the hypotheses you form. And that's how you're going to be able to explore worldwide questions with a little system like this.